All right, we're on to part 4, the last of chapter 6. Just a few other sort of miscellaneous items. So there's one aspect within cells, an interesting thing that it's important to see or understand, that the various organelles and structures within a cell are fluid, and they're constantly moving as they interact in chemical interactions, pull and push and so on. So one of those aspects is called cytoplasmic streaming, which is a type of motion that actually microfilaments enhance as part of this highway system. And if you look under a cell, under a microscope at a cell, a plant cell, you can actually physically see these chloroplasts moving around the cell. Like this. This is a quick video in real time of those chloroplasts. Now, of course, you can't see it green here, but those are the chloroplasts moving around. That's cytoplasmic streaming. The last one, intermediate filaments, they also help to support cell shape. Remember, they're part of the cytoskeleton. And they help to fix organelles in place, so that some of them we don't want moving and streaming, as you saw the chloroplast doing, such as the nucleus. We generally want to hold it in place. And then the relationship or the interaction with the ER and the Golgi will also largely be static. Cell walls of plants and other organisms, but mostly of plants is our focus right here. They're made of cellulose fibers, as well as some other proteins, of course, and they may have multiple layers, as you can potentially see. But cell walls are rigid, and they provide structure, and they also provide a support or a wall to adhere to for other cells, so that one cell can attach to another cell through the or with the cell wall. Now, animal cells don't have cell walls, but they have a very specialized exterior structure called the ECM, or extracellular matrix, that acts very much like a cell wall and provides structure. It's made up of a lot of glycoproteins, proteins, carbohydrates, and so on, that you can see here. Here's the membrane, of course. And the ECM provides a number of functions, including support, so just structural in nature, binding to other cells, creating a tissue. So a bunch of cells work together to make up a tissue, for example. Adhesion, that same thing where they bind to one another. Movement, the ECM enables movement from one cell to the other, and communication, and then also regulation, also controlling what comes in and out of the cell, and signaling with other cells. Last topic is Plasmodesmata is one type of intercellular junction. We'll get to the other types in a second. Gap junctions and desmosomes. But Plasmodesmata are attachments between plant cells within the cell wall. Here they create their channels or perforations in the cell wall, and they are pathways for nutrients to flow from one cell to the other, to eliminate the need to pass through the selectively permeable membrane just underneath the cell wall. In animal cells, there's other types of junctions. There's desmosomes, as I mentioned, gap junctions, and also tight junctions. So we can see all those here, and I'll briefly describe sort of the purpose or how they all work. Here's tight junctions. Here we have a desmosome. It looks a little bit hairy. Okay, and then we have gap junctions. So first off, tight junctions are, as the name suggests, meant to hold cells very tightly together, one against the other. So a typical example of this would be in the small intestine. You have cells one next to the other, and you want nutrients and, let's say, bacterial cells to have to go through the membrane of the small intestine. That way you can selectively choose what comes in from the intestinal lumen, or space, where all the food is, into the cells. If there were gaps between the cells, holes, then nutrients and bacteria, viruses, and so on could pass between the cells and make their way into the bloodstream. Tight junctions prevent that from happening and force everything to pass directly through membranes to get into the bloodstream. Desmosomes are sort of the generic facets that hold cells together to take a group of cells into a tissue. And then gap junctions, lastly, are the animal cell form of Plasmodesmata, where they are pores connecting two cells that allow ions and nutrients to pass from one cell to the other without having to once again pass through the semi-permeable membrane. Bottom line, cells rely on the integration of structures and organelles, and those all tie together to create that cell's function.